So I'm Luiz Sergio, working in Brapa. I'm also a young member of the Sustainable Bio. We started working with them in 2020. So we have here the Rubens and Maria Lucia. Also, they are a member, older than me in the Sustainable Bio, so they have much more experience than me. So I'm trying not to take much time here. So just to show you some data. Yesterday, you watched Maria Lucia and Fernando Garcia showing something about the Sustainable Bio, how it works here in Brazil. So this I don't want to repeat that because yesterday we saw that. Here just to give an idea of the GMO commercial products approved by Bio since '98 from 2021. So you have here a vegetal microorganism, animal therapy, human therapy, EGM animals. So you see just 3 animals here. And here is a number of, just to give an idea of how hard this all work in the Sustainable Bio. This is the number of the process. They are evaluated every year. So just for the last 4 years, so more than 1,000 process. So it's not only for products, but also annual reports from CE Bios and several others. Any project with activity with genetic engineering should be submitted to the Sustainable Bio. So because of that, we have a lot of work on Sustainable Bio. So my plan here is just to talk a little bit about the normative resolution number 16. So we have here yesterday also Fernando Garcia showed something about this normative resolution. So this normative resolution is to establish the technical requirements to present to Sustainable Bio a query or a consultation about the products generated by new breeding technologies. So here we're going to see if the product can be determined as non-GMO. So if it's not non-GMO, will not be under the Sustainable Bio regulation. But we have to keep in mind this still need to be regulated by other Brazilian agencies as a conventional product. So the main requirements are product without any introduction of exogenous or recombinant DNA or RNA. Product can have site-directed mutations for gain or loss or loss or function, but only without introduction of recombinant DNA RNA. So in this case, the SDN1 can be considered non-GMO. SDN2 can also be considered non-GMO since there is no introduction of any exogenous DNA. So you can use a template to introduce 1, 2, or 3 point mutations. So it's a little bit more precise than use only the knockout. So so in your case here, you can use SDN1, SDN2 approach to generate a product that can be considered non-GMO. So we need to show here the genetic engineering strategy used to edit the genome, and how on targets and off targets were detected and evaluated, the DNA sequence, the localization, the presence of targets, and the product and the gene. So here is just what we, the Sustainable Bio, request when someone wants to submit a consultation. So this is regarding the animal origin, the source of the parents. So identification of genetic technology, the purpose, the intended use, the risk classification, so the biosafety level, genetic strategy to produce the desired modification, the molecular characterization of the result of manipulation, number of manipulated copies, etc. The product of the extraction. You have also to prove the absence of recombinant DNA or RNA molecules. If the product of the request is commercially approved in other countries, this is also interesting. If someone submitted in another country was approved, here we also take this in consideration. So it's a part of the process. So if the product uses the principle of gene drive, you have to explain how the organism will be monitored, and you have to use at least 2 different strategies to check what's going on with your gene drive product. The possible unintended effects need also to be evaluated if present. So here, just to show you the number of consultations submitted to Certain Bio since 2018. So here's the number of the blue is submitted one and the red is approved. So just few, you can see this, we have just few. In 2021, we have a bigger number here, it's about 12. Most of them are microorganisms. We have some with plants. We have of animals. We have one vaccine and one mosquito. So actually, so most of the non-GMO products approved, considering non-GMO by Certain Bio, are microorganisms, yeast. Most of them are yeast to use in the biofuel production. In this case, ethanol. So we're gonna, the idea now is just to show you 3 examples of non-GMO. There was, product that was considered non-GMO by Certain Bio. One is the mosquito. Actually, it's not exactly the mosquito, but it's the product, the RNA product that can be used on mosquitoes. So you use the double-strand RNA that targets the tested genes. So this is the way it works. So the antisense RNA targets the complementary RNA, and then you have the adult male that greatly reduces fertility. So that product, so the RNAi to be used with mosquitoes, Aedes aegypti, were considered non-GMO in 2019. The other example is well known is the tilapia denial, tilapia with increased fillet yield. So the fish embryos were injected with CRISPR-Cas9 RNA to target the myostatin gene. So they have the founder. Then they have the first generation. They cross the first generation to have the second generation. So the second generation is homozygotes. So they have the FLT fish. So they have a deletion of nucleotides. So they were able to have a stop codon to have a truncated protein that have this kind of phenotype, knockouting the myostatin gene. The result was an increase in growth rate and feed conversion. And that was considered non-GMO in 2019 too. And here I have just, I put here just 2 examples. Tomorrow I'm supposed to talk a little bit more about cattle, genome-edited cattle. So I can give you more details about those 2 products considered non-GMO. But here we have the semen from a bull, the Lorry bull with double muscle. They use the Talens approach to generate those animals. So they introduced in the house to knock out the myostatin gene. And also you have the one male and it's not this one here, this is just example, but one male and female with sleek hair. So in that case, the approach was to using CRISPR-Cas9 injection, also in the IVF zygotes. So they were able to insert mutations in prolactin receptors. So both were considered non-GMO in 2021. So that was, I was trying to be faster, right? Not to take much time or few, but just to show you that we most of the products submitted to Certain Bio are microorganisms. We have a big industry of ethanol production here. So we have also sugarcane. There was also considered no GMO. Soybean also considered no GMO. So sugarcane with great biodigestibility and more sugar to produce ethanol. Soybean with low rephenols is anti-nutritional product in the soybean. So all of them using the CRISPR system. So that's it. So thank you for your patience.